Hi everyone, this is Miss Lasar, and today I'm going to be teaching you about meiosis. Wow, that sounds very similar to mitosis. There will definitely be some similarities, but they are two different processes. This is cell division, similar to mitosis, but with a different purpose, to form sex cells. Most important thing is during today's lecture, make sure you are taking those fabulous, detailed, handwritten notes. Let's do this. We've got 4 lesson questions to answer today. First question, what is meiosis and what makes it different from mitosis? Number 2, what are homologous chromosomes? Number 3, what happens during meiosis? And number 4, what can go wrong in meiosis? So we'll talk about purpose, we'll talk about some key vocab, we'll talk about the details of the steps, and then finally, what can go wrong. Let's focus in on question 1. What is meiosis and what makes it different from mitosis? Let's start with a review of mitosis. So you've seen this picture many times before. This shows you the process of cell division. You could probably talk it through in your sleep. We've got our cell with our duplicated DNA, nuclear membrane dissolves, DNA gets sent to opposite sides of the cell pinches apart, and now we have 2 identical daughter cells. This is mitosis. This is in black. Go ahead and write it down. Purpose of mitosis is to divide a somatic cell, that's a body cell, in half and end up with 2 genetically identical daughter cells. Key thing about mitosis, daughter cells have the same number of chromosomes to each other and have the same number of chromosomes to the parent cell. So daughter cells have the same amount of DNA and the same amount of DNA as compared to their parent cell. Let's contrast that with meiosis. Here is a picture showing meiosis. You do not need to copy down this picture. We'll go through the details of the steps in a little bit. This is a process that is involved in creating your sex cells. You can see an egg cell and a sperm cell in the second picture. And the purpose of meiosis is to divide one cell into 4 genetically different cells that each have half a set of DNA, only 1 copy of each chromosome. So we've seen a bunch of differences already. This is the process that produces your sex cells, the sperm and egg cell. Notice the differences from mitosis already. We start with 1 parent cell, but we end up with 4 daughter cells, not 2. We start off with 1 parent cell with 2 copies of each chromosome. We end up with 4 daughter cells that each have 1 copy of each chromosome, and all 4 cells are genetically distinct from each other. So definitely some differences from mitosis already. Dialing in on the purpose of meiosis, meiosis only happens in ovaries and testes. It does not happen anywhere in the body, so it only happens in specialized cells in your reproductive system. Here's another image showing you the differences and similarities between mitosis and meiosis. You do not need to copy down this image. It is just to compare and contrast. Notice that we start with 1 parent cell. These are the same. They both have 2 sets of each chromosome, and it looks pretty similar. We've got processes of cell division. We're pulling chromosomes apart. We're dividing into 2 daughter cells. But then notice that there's an extra row here in meiosis, and we divide our 2 daughter cells once more into 4 final daughter cells. Again, we'll go through these details shortly. Section 2, some vocabulary. What are homologous chromosomes? Let's back up and review what a chromosome is. Here's a picture of a chromosome. A chromosome is a condensed DNA molecule. So now you're going to learn a little bit more detail about that condensation. You previously learned that the DNA is condensed by wrapping it tightly over itself, kind of like winding a thread on a spool. And histones, you can actually see in this picture, histones are these little pink circles, and these are proteins that the DNA wraps around to form a condensed shape. This is in blue, so you don't have to write it down unless you think it's helpful. A good way to think about histones are that they are like the spool that thread would be wrapped around. So wrapping our DNA very tightly around our histones and then further coiling that back on itself forms the tightly woven, tightly wound condensed chromosome shape. Some more vocabulary. What you're looking at is a picture of chromosomes. This is called a karyotype. This is an image, this is actually a photograph that shows the entire set of chromosomes possessed by an individual. So this is a human karyotype, and what you can see in this karyotype are the 46 chromosomes that this individual has in every single one of their cells. Karyotypes are created by extracting chromosomes from the cell when it is in the process of cell division. So when the chromosomes are in this tightly wound shape, makes them much easier to visualize and sort. In each human cell, there are 46 chromosomes, and you can tell that they exist in pairs. So there are 23 pairs overall. And what you might notice is that they're organized by size. Chromosome pair number 1 is the largest, down to pair 22, which is the smallest. And then we have a funky pair over here. This final pair, the 23rd pair, these are your sex chromosomes. So this is a recap. You already wrote this down. In each human cell, there are 46 chromosomes in pairs, 23 pairs total. The last pair is the sex chromosomes. If that pair is two identical X's, then this is a female. If there is one X and one very small Y chromosome, then this is a male. Each pair is defined by the genes that they have on them. For example, all chromosome 4s have the same set of genes on them. They have the same set of genes in the same order down this chromosome and down the other chromosome 4. So, where did these pairs come from? Well, each pair is made of one chromosome from mom and one chromosome from dad. These are called homologous chromosomes. We've been referring to them as pairs. Now we're going to call them homologous chromosomes. These are the matching chromosomes that have the same set of genes. So if we look at chromosome pair number 2, we can see one chromosome, which we're inferring in blue is inherited from your dad, and one in in red, which is inherited from mom. Here's another chromosome pair. The blue is pointing to the one inherited from dad. The red is pointing to the one inherited from mom. If we look at chromosome 1, each homologous pair is defined by the genes that they have on them. By the way, this is in blue. You don't need to write this down unless this is helpful or interesting. Homologous pair number 1 has the same genes on them. Both homologous chromosomes can have different versions of their genes. So what does that mean? Well, first of all, it means you have 2 copies of every gene, but those 2 copies can be slightly different from each other. If we take a look at this homologous pair, we can see that they both have the gene for eye color right here at the top, and then they've got a copy of the gene that determines blood type, and then they've got a copy of the gene that determines hair color, and then they've got a copy of the gene that might determine growth. So we call each copy of a particular gene an allele. An allele is a different version of the same gene. So you have 2 alleles for eye color, 2 alleles for blood type, 2 alleles for hair color, and 2 alleles to determine your stature. You have 2 alleles of each gene, one on each chromosome that gene is found on. There are genes that code for eye color on each homologous chromosome, but there can be slightly different eye color versions coded on each. For example, your mom might pass down an allele for brown eyes, and your dad might pass down an allele for blue eyes. We're moving on to section 3, the details. What happens during meiosis? This is going to be represented by a series of steps. Make sure you write down the text of every step. The pictures are optional, but you may find it helpful to draw a representative picture for each step. There are going to be a lot of steps. Meiosis is much longer than mitosis, so we're going to end up with something like 15 or 16 steps overall. And just like in mitosis, we're actually going to start thinking about meiosis before cell division, way back in S phase. I'm starting with a cell in S phase. This is the beginning of S phase, and I know that because it has non-duplicated DNA. There's just one copy of each chromosome. By the end of S phase, I'll have gone through DNA replication, and I will have 2 sets of DNA. I'm now at the end of S phase with my duplicated DNA. And meiosis will start, number 1, once the nuclear membrane starts to dissolve. Meiosis has begun. So you can see that my nuclear membrane has disappeared, and there are some other things that have appeared. Perhaps you remember what they are. Those are centrioles. Step 2, the chromosomes will condense. So I go from this loose chromatin form to tightly wound X-shape chromosome form. And notice how many pairs I have in here. I have, click so that you can see them. There we go. I've got 2 pairs of the large chromosome, 2 pairs of 2 copies of the medium chromosome and 2 copies of the small chromosome. So overall, I have 3 pairs of chromosomes in here, and I've color-coded them: blue from dad, red from mom, and let's keep an eye on those pairs. So notice this is not representative of a full human cell, because in a full human cell, I would have 23 pairs, but that was very hard for me to squish in on one slide, so I went with 3 pairs, which was much easier to represent. We're ready for step 3. In step 3, the centrioles move to opposite sides of the cell. There they go. Step 4, the homologous chromosomes, remember those are the matching pairs. Homologous chromosomes physically pair up. Let's watch them do that and watch how they align in the cell. So my medium-sized chromosomes have paired up side by side. My large chromosomes have paired up side by side, and my small chromosomes have paired up side by side. What you might want to draw in your notes right now is a picture of homologous chromosomes paired up side by side. Notice how they are overlapping. Notice where they're touching. It's 2 X's side by side physically touching each other. And that's really important, because now something crazy is going to happen. And that something crazy is called crossing over. Crossing over is when homologous chromosomes physically swap pieces. Let me show you what that looks like. We start with our homologous chromosomes aligned, and then they actually pair up next to each other as you've drawn in your notebook. And notice how the blue leg is overlapping over the red leg, and what might happen if they twist a little too tightly? Well, those chunks are actually going to swap sides. And now we've got a blue chromosome that's got a red chunk on the bottom, and a red chromosome that's got a blue chunk on the bottom. And that means that genetic material from dad and genetic material from mom have kind of swapped places, and we end up with what's called recombinant chromatids made of some genetic material from one parent and some genetic material from the other parent, which is totally crazy. We also have on the outsides those fully red chromatids or fully blue chromatids, and those are our non-recombinant chromatids, where the genetic material is just from one parent. What I would recommend doing is, in your notebook, drawing this process. Draw your homologous chromosomes aligning or lining up next to each other, draw them overlapping, draw them swapping sides, label them, shade one in a different way so you can tell that they've actually swapped genetic material with each other. And now we go back to numbering our steps of meiosis. We're on step 5. Step 5, the paired homologous chromosomes cross over their genetic material. Watch it happen. Bam! They've switched and swapped some chunks. I had them swap a piece on the top and a piece on the bottom so that each chromatid actually has 2 swapped chunks. It could be 1 swapped chunk, it could be 2 swapped chunks. Either way will happen, and it's kind of random what ends up crossing over and what doesn't end up crossing over. Step 6, the centrioles, thought we forgot about them, begin making spindle fibers. There they go. And that leads us to step 7. The spindle fibers push the homologous chromosome pairs to the center of the cell. So notice our homologous chromosomes are still physically paired up with each other, and the spindle fibers are just going to push them to the center of the cell. This looks a lot like metaphase in mitosis, and although we're not making you learn the names of these steps, this is actually called metaphase 1 of meiosis. It's a very similar construction. We have that line of chromosomes down the center of the cell, but right now they're actually paired up with each other, whereas in metaphase in mitosis, it's a single line of chromosomes down the center of the cell. Step 8, the spindle fibers shorten and pull the homologous chromosomes apart. This is going to sound a lot and look a lot like anaphase in mitosis. Watch them go. They're separating, they're being pulled apart just like in anaphase. But in anaphase of mitosis, those would be sister chromatids that are being separated from each other. And right now, notice that they are homologous chromosomes. It's a whole duplicated chromosome that's being pulled apart. Step 9, eventually the homologous chromosomes are totally separated. There they are. Step 10, the nuclear membrane will reform around them and the cell will start to divide. I know this cell is a little funky looking, but notice we've got our nuclear membrane around each set of homologous chromosomes, and our cell is starting to pinch in the middle. It will divide into Two cells. Step 11, two cells are formed. They have one copy of each chromosome, and the chromosomes are duplicated. So we still have those X shapes. We still have a duplicated chromosome with two sister chromatids. One of the chromatids is recombinant, the other is not recombinant, and we don't have two of each chromosome, we have one of each chromosome in each cell. Step 12. The nuclear membrane will dissolve again. Centrioles will form again and move to opposite sides of the cell again. There they go. Step 13, spindle fibers will push the duplicated chromosomes to the middle of the cell. Spindle fibers, push, push, push. Whoa, look at this. This really seems like we repeated steps, and we kind of did. Now this looks a ton like metaphase in mitosis, where we have our duplicated chromosomes in a line down the center of the cell, attached on both sides to spindle fibers. You can infer what's going to happen next. Step 14, the spindle fibers shorten and separate the sister chromatids. Bam, they get pulled apart. Wow, just like anaphase. And this is such a repeat of what you already saw in the first part of meiosis. We did exactly this, but we pulled apart the homologous chromosomes. And now we're repeating it and pulling apart the sister chromatids. Step 15, the sister chromatids are fully separated. Let's send them to opposite sides of the cell. There they are. Step 16, the nuclear membrane reforms. So we've got our nuclear membranes forming around both groups of sister chromatids, and step 17, our cells will divide. There we go. We pinch and our cells divide. Now we have 4 daughter cells. Just by looking at the picture, you can tell that the chromosomes in each daughter cell are not the same. Look at their colors. Look at whether they're recombinant or not recombinant. This is step 18, our final step. We end up with 4 sex cells. They're either 4 egg cells or 4 sperm cells, depending on which reproductive system they are forming in. Let's compare. On the left, we have our original cell. This original cell is much larger. It has 2 copies of every chromosome. On the right, we have our daughter cell. It is much smaller, and there's only 1 copy of each chromosome, and some of those copies might be recombinant. We have one other thing to talk about in meiosis, and this is going to make things even more complex. It's called the law of independent assortment. You might have been wondering how we ended up with the combination of chromatids in our final cells that we did. It's because of this law. This law states maternal and paternal, so coming from your mom and coming from your dad, chromosomes get divided randomly between sex cells. Maternal and paternal chromosomes get divided randomly between sex cells. Let me show you what I mean by this. Here's our starting parent cell. The alignment of the homologous chromosomes down the center of the cell will determine which chromosomes end up in our final cells. For example, if I take these homologous chromosomes and I divide them apart, I'm going to end up with 2 sex cells formed from this combo on the left, 2 chromosomes from dad, 1 chromosome from mom, and another 2 daughter cells that will end up with this combo, 2 chromosomes from mom and 1 from dad. And that just has to do with the way these chromosomes are lined up down the center of the cell, where I've got these 2 red ones on the right and then a blue one. That's getting sent to 2 daughter cells. And I have 2 blue ones on the left and a red one. Those are getting sent to the other 2 daughter cells. What if the lineup looked like this? Well, then when I separate these homologous chromosomes, I'm going to end up forming 2 sex cells that have all chromosomes from mom and 2 sex cells that have all chromosomes from dad. But it's even more complicated because remember what happens when our homologous chromosomes are lined up in the center like this? They swap genetic material, they cross over, and we actually have chromosomes that look like this that are recombinant. And so my complicated recombinant chromosomes on the left will end up forming a set of complicated genetically different sex cells inheriting this info. And the sex cells that I end up with from the right will inherit this different set of recombinant or not recombinant genetic info. So what's even more complicated is that chromosomes are usually slightly mixed because of crossing over. Let's look at an example. I think this will make it helpful. So right here, I've got 2 chromosomes. I'm calling them chromosome 1, the big one, chromosome 2, the little one, and let's say these are your chromosomes. Dad gave you the genes for, okay, here's our color key, and what's being represented here by the stripes is the genes that are on each chromosome. So chromosome 1 has the gene for cleft chin and the gene for widow's peak. Cleft chin is like a bump in your chin, and widow's peak is a triangle at the front of your hairline. Chromosome 2 has the genes for dimples and the genes for freckles. Dimples are little indentations in your cheeks, and freckles are little dots on your skin. So, if we're looking at the blue chromosomes, you inherited these 2 blue chromosomes from your dad. Your dad gave you the genes for dimples. That's what, where's dimples? Oh, it is dark gray, right down here on chromosome 2. There's dimples. Freckles, that's dark green. No cleft chin, that's this light blue stripe right here, and no widow's peak, that's this light yellow stripe here. So those are the genes that you inherited from your dad. From your mom, you inherited the genes for no dimples. So check, that is light gray, the gene for no dimples, the gene for no freckles in light green, the gene for cleft chin in dark blue, and then the gene for widow's peak in dark yellow. So these are the genes that you have in all of your cells, but the question in meiosis is not what do you have? The question is what will you pass on to your children? Because remember, you're only going to pass on 1 copy of chromosome 1 and 1 copy of chromosome 2. So which one will you be passing on? Well, the law of independent assortment says that you could pass on either one of chromosome 1 and either one of chromosome 2. So you could pass on this copy of chromosome 1 and this copy of chromosome 2, or you could pass on both of the maternal ones, this one from chromosome 1 and this one from chromosome 2. And then these other 2 chromosomes just never get passed on. You have that info, but you don't pass it on to your kids. Crossing over means that there will be some recombination between the 2 chromosome 1s and between the 2 chromosome 2s. So let's visualize that. Let's look at our duplicated homologous chromosomes. It's helpful to look at them as they're duplicated so that we can really envision what happens during crossing over. And let's make a little crossing over happen. I swapped the top arms of chromosome 1 and the bottom arms of chromosome 2. And now you can pass on new combinations of genes that we haven't seen before. So, for example, you could pass on the gene for cleft chin, widow's peak, dimples, and no freckles. We've actually never seen this combo of dimples and no freckles before because this is half from your dad and half from your mom. Or you could pass on this non-recombinant chromatid from your dad and this recombinant chromatid made of your mom and your dad's genetic info. There's so many possibilities. We could pass on still 2 non-recombinant chromatids, just the genetic info that you inherited from your mom. The law of crossing over plus independent assortment leads to major genetic variation in all egg and sperm cells. This might explain things if you have ever wondered why you and your siblings look so different. Because even though you may have the same parents and they have their own genetic information, they're passing on different combinations of genes from their parents onto you. And because those combinations are so varied and happen randomly, you and your siblings inherit different combinations of genetic info, and you all look different. Okay, final section. What can go wrong in meiosis? Well, there's one big thing that can happen. It is called non-disjunction. This is failure of homologous chromosomes to separate during the first part of meiosis. And here's what it looks like. Here's our parent cell, and I've got my homologous chromosomes already lined up. And what I know should happen is I should end up with 2 daughter cells where there's the chromosome 1 pair that has separated successfully, and I know my chromosome 2 pair should separate. But it doesn't. For some reason, there's some spindle fiber failure, and we end up not separating this pair of homologous chromosomes, and they both get sent to the same cell. We can already see that there's a major issue here because the cell on the left has 2 copies of chromosome 2. The cell on the right has no copies of chromosome 2. Oi. As we continue through meiosis, the sister chromatids will now separate, so we'll end up with 2 daughter cells forming from each. On the right, our sister chromatids separate, and this is all they end up with. They just end up with one copy of chromosome 1, nothing of chromosome 2. On the left, chromosome 1 sister chromatids separate, chromosome 2 sister chromatids separate. So on the left, we now have 3 chromosomes where we should have 2 chromosomes, one copy of chromosome 1 and 2 copies of chromosome 2. This journey is not over because we kind of need to think about what's going to happen after fertilization. So fertilization is when egg and sperm combine, and that's when we're going to add another copy of each of these. Let's imagine these are egg cells that we've just formed, and we're going to combine them with the genetic info from a sperm cell. Well, they're all going to get one copy of chromosome 1 and one copy of chromosome 2 from the sperm cell. But now if we look in our final fertilized eggs, on the left, our 2 left ones, we have 2 copies of chromosome 1 and 3 copies of chromosome 2. On the right, we've got 2 copies of chromosome 1 and only 1 copy of chromosome 2. In all of these cases, we have formed cells that do not have the right amount of genetic information. One thing to know is that while this actually happens fairly commonly, it almost never produces a human that can survive to birth. This is almost always fatal to a developing embryo. There are very few circumstances where having an extra chromosome or missing a chromosome is actually survivable. And I'm going to tell you about 3 of those situations now. This results in some cells having 3 or 1 copy of a chromosome instead of 2 copies of each chromosome. Our first genetic disease caused by non-disjunction is Down syndrome, which you may have heard of. This is a disorder resulting from non-disjunction of chromosome 21. And to develop Down syndrome, you end up with 3 copies of chromosome, that should say 21, 3 copies of chromosome 21. If you look on the karyotype, the picture of the chromosomes, let me focus you in on this blue circle, you can see 3 chromosome 21s. The symptoms of Down syndrome are quite severe. This is any extra chromosome or missing chromosome is going to be a major impactor health-wise. Intellectual disabilities, language delays, many different facial differences and physical differences. Facial differences include a flattened face, a small head, a short neck, protruding tongue, upward slanting eyelids, unusually shaped or small ears, poor muscle tone, and excessive flexibility and short height. So Down syndrome is caused by an extra chromosome 21, and it's one of the fewest diseases that can be caused by an extra chromosome and still be survived. And that's actually because chromosome 21 is so small that it is possible to survive with 3 copies of it. Some of the larger chromosomes, like chromosome 1, 2, or 3, if you had an extra copy of those large ones, there's so much extra genetic material and you're making so many extra proteins that you shouldn't be making that those are much less likely to be survivable. Turner syndrome is another disease of non-disjunction, and this is a disorder resulting from non-disjunction of sex chromosomes, and an individual with Turner syndrome would have 1 X instead of 2 X's. So if you look at the karyotype, it's a little funky. We see 1 X instead of 2 X's or an X and a Y. So they only have 1 X chromosome as the entirety of their sex chromosomes. This is a survivable disorder, and individuals with Turner syndrome typically survive to adulthood and have normal lifespans. They are phenotypically, so by appearance, female, and they have slowed growth. Their adult height is significantly less than might be expected for a female member of the family. Failure to begin sexual changes expected during puberty. So you would not see an individual with Turner syndrome go through the normal changes that happen during puberty. Sexual development that stalls during teenage years, and an early end to menstrual cycles, not due to pregnancy. So what you're noticing is that without an extra X chromosome, without the second X chromosome, you're seeing infertility and you're seeing lack of proper development of reproductive organs and reproductive processes that impact overall growth. For most females with Turner syndrome, the inability to conceive a child without fertility treatments. And our final disorder of non-disjunction is called Klinefelter syndrome. This is a disorder resulting from non-disjunction of sex chromosomes. Again, an individual with Klinefelters ends up with XXY instead of XY for their sex chromosomes. So looking at their karyotype, again, we have a difference from the normal pattern. We see two X chromosomes and we also see a Y chromosome. Normally, we would expect to see two X chromosomes or one X and one Y, and here we have two X's and a Y. Symptoms of Klinefelter syndrome are infertility, a low sperm count or no sperm count. So, what you might notice is that we're referring to sperm here. An individual with XXY as their sex chromosomes is going to be presenting as by appearance male. They will have a small testicle and penis, a low sex drive, they'll be taller than average height, they'll have weak bones, decreased facial and body hair, less muscular compared with other men, enlarged breast tissue, and increased belly fat. So, just like Turner syndrome, we're seeing impacts over the whole body, including both the reproductive systems and overall growth and development. So, these are three disorders that result from non-disjunction, the primary thing that can go wrong in meiosis. I hope that you have learned a lot from today's lecture, and we're excited to practice this all with you in class. Thanks for watching.